Good afternoon, everyone, and welcome to today's class. We're gonna start in 5 minutes so that more people can join in. Okay, let me share my screen with you guys. Yeah, yeah, I hope you can see it. Okay, so we gonna have a short class today. It's a small topic which I wanted you guys to be familiar with. As you know, we are talking and discussing about membrane proteins, how they function, then the protein part of it. Another important aspect of membrane protein, as I have told you, the function which involves membrane protein includes transportation of ions across the membrane, right? And also we have seen the action potential and the potential energy associated with the membranes because of the differential distribution of the charges or electrolytes in this case. Okay, but have you thought that how exactly we measure it? When we are saying that potassium is more inside, sodium is more outside, for chlorine, for calcium, for all these small ions, and there is a ratio which needs to be maintained so that the membrane can stay in the exact potential which it is supposed to be, right? So to maintain this, the proteins helps in transportation of these ions via channels, transporters, and all the proteins which are at the end of the day present in the membrane. So how do we actually measure it experimentally? How to know it, that what is the exact concentration of ions, right? And how a channel or a transporter which is involved in the transportation of particular type of molecule is transporting at what rate, correct? So you know, whenever we talk about movement, for example, a car is moving, any vehicle is moving, you are moving, anything which is moving, we like to understand its rate. Rate means any quantity with respect to time. So similar thing is with channels and transporters. We know there are n number of channels, we know there are n number of transporters, they transport in various different ways, right? But how effectively or efficiently they are transporting? You remember when we studied about action potential, we said there are some fast channels, some slow channels, right? So what do we exactly mean by fast channel or slow channels or leaky channels, right? It basically means that the rate at which they are transporting their respective substrate. So that rate can only be measured with respect to time. So we should have something in our hand which can actually determine that with respect to time, how many solutes or how much substrate is being transported. That is what basically electrophysiology is. Okay, so electrophysiology involves measurements of voltage changes or electric current or manipulations on a wide variety of scales, from single ion channel proteins to whole organs like the heart. In neuroscience, it includes measurement of the electrical activity of neurons and, in particular, action potential activity. Recordings of large-scale electric signals from the nervous system, such as electroencephalography, may also be referred to as electrophysiological recordings. They are useful for electrodiagnosis and monitoring. Okay, so before discussing much into it, let us go back to the introductory video for this particular electrophysiology. Let me just share the screen.
Hey, how much time would it take you?
Yeah, 1 minute. I have muted because it's an ad, so I'll hope that you guys can see it. Yeah.
Welcome to another physio video. Today let's talk about electricity in cells. The study of how cells conduct electricity is known as electrophysiology. This video is part 1 and discusses the resting membrane potential and the action potential. Think of your senses. Sensory receptors located throughout your body will transduce different forms of energy like light waves or sound waves or mechanical energy from touch into electricity that can be transmitted by neurons to the brain. An electrical signal sent toward the central nervous system is known as an afferent signal. The brain receives afferent electrical signals and interprets them as important information about our internal and external environment. the brain also sends electrical signals out to control muscles and glands. Additionally, our thoughts and personalities are all possible because of the electrical capabilities of our brains. Electrical signals traveling from the brain to effectors such as muscles to cause contraction for movement and to glands for secretion are known as efferent signals. Hopefully you can see how important electrical signals transmitted along neurons are for both sensory and motor functions in the body. But what about neurons makes it possible for them to transmit electricity? Let's take a neuron and blow up part of it so that we can see how the proteins embedded in its plasma membrane make this possible. The main players that make electricity possible are potassium leak channels, sodium-potassium pumps, negatively charged proteins, sodium voltage-gated channels, and potassium voltage-gated channels. Let's talk about how each of these is important for sending electrical signals. If we position an oscilloscope or voltmeter with the recording electrode inside the cell and the reference electrode outside the cell, a potential difference of about minus 70 millivolts is recorded for the neuron. It is a negative number because the inside of the cell is negative compared to the outside of the cell. The inside being negative is an important characteristic that allows neurons to send electrical signals. Three main players are involved in making the inside of the cell negative. First, negatively charged proteins are made inside the cell and become trapped, making the inside more negative. Secondly, the sodium-potassium pump uses ATP to pump 3 sodium ions out of the cell for every 2 potassium ions it pumps into the cell. This pump moves more positive ions out of the cell compared to positive ions brought into the cell. This imbalance of charge movement due to the pump contributes about 15 millivolts of the total 70 millivolt potential difference. The sodium-potassium pump also creates gradients. Since sodium is pumped out, the concentration for sodium becomes higher outside the cell compared to inside the cell. Potassium is pumped into the cell, so the concentration for potassium becomes higher inside the cell. In addition to the negatively charged proteins and sodium-potassium pumps, leak channels for potassium are also very important to allow the inside of the cell to be negative compared to the outside. Since potassium is higher inside compared to outside, it will diffuse from high to low down a chemical gradient. Remember that potassium is a positive ion, and the more that goes down its chemical gradient and leaves the cell, the more negative the inside of the cell will become. The inside becoming more negative will attract more of the positive potassium ion back into the cell. The attraction back into the cell is due to an electrical gradient. Together, both gradients form the electrochemical gradient. They are opposing forces. One is a force out of the cell, and the other is a force into the cell. When the amount of potassium leaving is equal to that coming into the cell, equilibrium is reached, and this is known as the resting membrane potential, or RMP. The RMP is approximately minus 70 millivolts for neurons. Do you recall the 3 main players that allow the inside of the cell to be negative and establish the resting membrane potential? They are negatively charged proteins, the sodium-potassium pumps, and the potassium leak channels. Now let's talk about voltage-gated channels. These channels include the sodium voltage-gated channels, and the potassium voltage-gated channels. These channels allow an electrical signal to travel the length of the neuron. We call these traveling electrical signals action potentials. The sodium voltage-gated channels have two gates, an activation gate on the outside of the cell membrane and an inactivation gate on the inside of the membrane. Potassium voltage-gated channels just have one gate, located on the outside of the cell membrane. How do the voltage gates open to generate an action potential? Here's an example of how an action potential may be triggered. You are examining Santa Claus's sore back. When you touch his back, you have applied a mechanical stimulus to touch receptors in the skin on Santa's back. The stimulus increases the permeability of the membrane to certain cations like calcium. Calcium will go from high to low and rush into the cell. The neuron was resting at -70, but now, since calcium came into the cell and calcium has a positive charge, the RMP will go up a little bit. If the stimulus is large enough to reach threshold, sodium voltage-gated channels will open, bringing about an action potential that will travel the length of the neuron. Also note the graphical representation of the action potential. When the signal makes it to Santa's brain, he becomes consciously aware he is being touched.
People say women are emotional.
So when the cell is at rest and not sending signals, the RMP has been established by sodium-potassium pumps, leak channels, and negatively charged proteins. RMP simply means that the inside of the cell is negative at rest. And notice that the sodium and potassium voltage gates are closed at rest. Remember that in order to get an action potential, first we must have an excitatory stimulus that will bring the membrane potential to threshold. The example we gave was a mechanical stimulus that increased calcium permeability, allowing calcium to enter the cell and make the inside of the cell more positive, allowing threshold to be reached. This stimulus is called an excitatory graded potential. When we get to threshold, the activation gate on the sodium channel opens, and the potassium voltage gate starts to open, but opens slowly. Fully open sodium channels will allow much more sodium to enter the cell compared to the amount of potassium that is allowed to leave. The positively charged sodium ions entering the cell will make the membrane potential more positive. This phase on the action potential graph is called the depolarization phase. At around 0, near the end of the depolarization phase, the inactivation gate closes so sodium can no longer enter the cell. By this time, the potassium voltage gate is fully open, and lots of potassium goes down its gradient and leaves the cell. As the positively charged potassium leaves the cell, the membrane potential becomes more negative. This phase of the action potential is called the repolarization phase. After repolarization, sodium voltage gates have returned to their original configuration that they had at rest. The activation gate is now closed and the inactivation gate is open. Hyperpolarization is the inside of the cell becoming even more negative than -70 millivolts due to the potassium channels closing slowly. After the potassium voltage gated channel is completely closed, the cell returns to minus 70 millivolts resting membrane potential. Once again, the cell is only permeable to potassium through the potassium leak channels, and the sodium-potassium pump is always working to maintain the sodium and potassium gradients. A gradient means that there is an area where it is high and where it is low. Please remember the potassium is high inside the cell and sodium is high outside the cell. In summary, please be able to answer two questions. First, what three main things bring about the resting membrane potential? If you answered negatively charged proteins, sodium-potassium pumps, and potassium leak channels, you are correct. Second, what two main channels allow for an action potential? If you answered sodium voltage gated channels and potassium voltage gated channels, you are correct. This concludes.
Right, so this is basically why I wanted you guys to revisit this thing, is so that you know what we're gonna study next, right? So electrophysiology, as I told you, it is the study of voltage change or electric current across a membrane, or how it is transporting. So till now we have seen that how action potential is generated across the membrane at the end of the day, but that question still remains same: how do we actually measure it, right? When we are saying, okay, there are sodium potassium channels, there are sodium channels, there are potassium channels, they open, they are fast, they are slow, they transport 3 sodium or 2 potassium respectively, right? So all this information has to come from some experimentations, correct? So one such technique which helps us in understanding how the membrane proteins are transporting ions is called patch clamp. Okay, so let me show you this introductory video to patch clamp. With this you will understand what I mean by patch clamp, what is the name suggest, and also how do we measure the electric current across the membrane, across the biological membrane. Okay,
Just like we communicate with each other using cell phone, WhatsApp, etc., neurons in our brain communicate with each other by an electrical signal known as action potential or spikes. Now, if you're watching this video, the visual input is going into your brain, making your neurons of your eyes, the optical tract, to fire, and that information is relayed to the higher cortex where the visual information is processed. And if you're also listening to me, then your auditory neurons are firing and sending this information to your auditory cortex. So neurons spikes, that means the spike is action potential, which is nothing but a change in voltage over time. So the change is from resting membrane potential to a positive membrane potential of plus 30 millivolt. This delta change in voltage, about 100 millivolt change in voltage, is known as the action potential. So question is whether we can measure the action potential from the neurons or not. It turns out we can use electrophysiological methods to measure the action potentials, and that could tell us about various biophysical properties of the neuron, including capacitance, conductance, firing frequency, input resistance, and firing pattern. A combination of all of these biophysical parameters can tell about several aspects of the neuronal physiology and how the neuron is coding for the information, which would be decoded in the higher brain center. Now, here is a typical patch clamp setup with a microscope, and you can see with the microscope there is a camera attached by which you can real-time image the neuron from where you are recording. There are setups for electrode, stimulating and recording electrode, which are held in position by micromanipulators, and you have a bunch of amplifiers, several power supplies, etc., and a screen where the recording is displayed. So here is an even closer view of that rig, where you can see the micromanipulators is holding the patch electrodes, So these electrodes are recording electrodes. You could have from another side, you could have a stimulating electrode which would give current inputs. So now this is the stripped down version of the patch clamp setup. So most important part is this recording electrode, which is connected to the amplifier, because these currents or voltage, whatever it is recorded, is minuscule, so it has to be amplified in order to detect, and that goes to a data acquisition system and goes to a computer which displays the recording in real time. Now, the first and foremost criteria of the patch clamp is to establish a patch. So how to establish a patch? In a patch clamp method, the electrode is poured down towards the cell membrane and gentle suction is applied. Once the suction is proper, the membrane resistance would increase and reach in order of 1 gigaohm. We call it gigaohm seal. And let's see how to understand whether gigaohm seal is established or not. This seal is important because if its seal is not proper, then there would be ion leakage and the proper recording won't be happening. So first of all, when you are approaching with your electrode towards a neuron, you use a positive pressure and you inject a small current. So you would definitely get a linear response, a small change in voltage as well. Now what you could do, when you just reach and about to establish a gigaohm seal, then you inject the same amount of current. If the gigaohm seal or resistance is so high in order of gigaohm, then the recorded voltage deflection would be very, very low because your resistance is in order of gigaohms. Now gentle suction can break open the membrane of the cell, and thereby you would see a small capacitive transients which results due to the discharging of the membrane capacitor. So, and thereby you can understand a proper gigaohm seal was established, and now the inside of the pipette and the inside of the cell is continuous. This configuration is one of the important configuration and most used configuration in electrophysiology, known as whole cell recording.
Should you use Canva or PowerPoint?
Talk about whole cell recording, which is a subgroup of patch clamp techniques, then whole cell recording could be used or operated in two modes. One is voltage clamp mode, another is current clamp mode. Each has its own unique advantages. Using a voltage clamp mode, you can keep the voltage constant. So the voltage parameter is in your hand. You can keep the voltage constant, or in other terms, you can hold the voltage at certain millivolts and measure the currents. Whereas in the current clamp mode, you can keep the current constant and measure the change in voltages. So when you need to record the action potentials, which is basically delta change in voltage, you use a current clamp configuration. So a current clamp mode under whole cell recording. So, and when you need to record the currents, the ionic currents, then voltage clamp method is important. Now, voltage clamp method, or this overall patch clamp method, has several variants. So let me discuss briefly about them. So first of all, one variant is called as cell attached recording. When you establish the gigaohm seal but doesn't break the membrane, so single macroscopic and microscopic currents could be recorded in this configuration. The second configuration is the whole cell configuration, and most used configuration, where the inside of the pipette and the inside of the cell membrane is in equilibrium and they are like continuous. So in this configuration, we can record properties like action potential and macroscopic currents, etc., etc. Now, two other variants which are pretty interesting and important for specific application. For example, inside-out recording. In inside-out recording, the inside-out recording configuration could be achieved by simply pulling your pipette apart when the gigaohm seal is present. So that would create a patch of the membrane to be detached from the neuron. Now, since the inside of the ion channels is now exposed to the environment, you can study the properties of several molecules and how they regulate this channel conductance. So that is the utility of this inside-out recording. Now, the outside-out recording is another important thing. So in a whole cell configuration, if you pull the pipette backward, then a portion of the membrane is going to be retracted and it would be rejoined, and now the outside of the ion channels or outside of the membrane is exposed to the environment. Now that could be dipped into several solutions. Maybe this kind of configuration would be very useful to study role of several neurotransmitter on channel conductances. So all these kind of several variant of patch clamp techniques has its own advantages and disadvantages. But one of the most used one is the wholesale patch clamp recording that can be operated in either voltage clamp mode or in current clamp mode. Now, let's say in a wholesale configuration, you inject a negative current and you would get first capacitive small currents, and when it reaches the threshold, it would start firing action potential. And you can nicely represent this data in form of injected current and voltage and plot a IV relationship. Remember, when you are going towards more depolarizing, you are giving more depolarizing current, it is giving rise to your action potential. But why do you want to inject a hyperpolarized current? It is important to step increase the current, because in the hyperpolarizing side, you can calculate one important biophysical property known as input resistance, which would tell you how much leakiness is in the membrane, or how and also that parameter is important because depending upon input resistance, it tells us that how much the neuron is likely to fire, or how easy for the neuron is to generate an action potential. So these parameters I would be discussing in other videos. So, but what is the importance of this action potential? It turns out that our brain is a decoder of these action informations inside these action potentials. And it turns out, let's say a spider is trying to bite you and you are getting freaked out and your fear neurons are firing like hell. So after the time, there could be changes inside these neurons, which would change in biophysical properties of the neuron or conductive properties of the neuron. Let's say you've seen the spider for the second time and somebody is able to record from your brain. Then you would see a potentiated response. Your whole firing rate versus input current curve would shift leftward. In simplest word, that same amount of current is now given a heightened response or a potentiated response. So that's why these properties about action potential, firing frequency, firing rate can tell us so much about how our brain can function. Not only that, it turns out the information about sensory environment is decoded by the firing rate inside our brain. For example, the information about the odor intensity or odor quality which is present in our environment is actually decoded by the olfactory neurons in the higher brain center neurons by its firing rate. So firing rate and firing pattern in real time. It turns out the odors which are very similar have very similar firing trains. So that is interesting. So not only the amplitude of action potential, but the frequency of the action potential, the interspike interval of the action potential carry a huge amount of information about neuronal coding. So that is pretty interesting. So if you like my video, give it a quick thumbs up. Don't forget to like, share and subscribe, and keep on watching these whole video series where I would be describing several
Right? So now I guess you guys have an idea about a patch clamp technique which is used for actually measuring the current across the membrane. We'll dig into more theoretical aspect of it in the next class, which will be on Tuesday as scheduled always. Okay, thank you so much for your attention, and feel free to ask any questions if you have. Thank you.